In the millions of cells that make up a human body, have you ever thought that cancer can develop practically anywhere? Human cells goes through a process of cell division among themselves to create new cells as the body requires. Cancers are formed as a modification in genetic activity that result in a loss of control of cell behavior, causing uncontrollable growth of single somatic cell. Cancer cells invade normal tissues and organs and eventually spread throughout the body. Errors which occur during cell division, inherited, and environmental factors can result in changes that lead to cancer.
DNA is the template for life, so what happens when there are changes to this sequence? Substitution mutations occur when one nucleotide is replaced, but what effect can this have on the protein produced? There is more than one codon for amino acids, so some mutations, called silent mutations, don't alter the amino acid coded for. Missense mutations occur when the amino acid coded for is changed. Nonsense mutations involve terminating the transcription process entirely, producing an incomplete protein. Insertion and deletion of nucleotides can cause a frame shift mutation, where the entire subsequent portion of the gene is affected and different amino acids are coded for. So what does this mean for cancer? Lynch syndrome, a germline mutation inherited in an autosomal dominant manner, is primarily associated with colorectal cancers. This syndrome arises due to mutations in DNA mismatch repair genes that normally code for proteins that repair errors from DNA replication. Furthermore, 3' deletions of exons to the EpCAM gene can reduce the expression of the neighboring MSH2 gene via promoter hypermethylation. Another cancer that can be inherited is breast cancer.
Breast cancer is a common type of cancer that affects both men and women and can be caused by a variety of factors including age, family history, genetics, and smoking. The proto-oncogenes BRCA1, BRCA2, and HER2 mutate into oncogenes, which causes cells to divide and multiply abnormally. BRCA1 and BRCA2 genes are located on chromosomes 17 and 13. These genes produce proteins that inhibit tumor growth and repair any DNA damage. This is a result of germline mutation which develops in gametes and can be passed down to the offspring at the time of conception. HER2 gene is also located on chromosome 17 and codes for a protein that promotes the growth of cancer cells. However, HER2 genes are caused by somatic or acquired mutations and cannot be inherited by the offspring, rather caused by external factors like lifestyle or environment.
Out of the women diagnosed with ovarian cancer, only a few have been hereditary. However, a family history of cancer does increase the chances of inheriting mutated genes and develop into a cancer. Germline mutations like BRCA1 gene, primarily known as the breast cancer gene, can also be responsible for developing ovarian cancer. So before the cells get a green light into the cell cycle, they have to pass each checkpoint or get stopped when mutations are detected. Single-stranded DNA breaks, which occur on one strand of DNA, much more simple to fix compared to the double-stranded DNA breaks, which is broken on both strands, requiring an allied identical DNA region to utilize as templates and reconstruct the broken regions. This is the homologous recombination repair mechanism at work. Mutations activate DNA repair genes like BRCA1 to recruit proteins like RAD15 on site to mediate strand exchange. What if DNA repair genes are damaged themselves or not activated? To repair the proto-oncogenes or the DNA repair genes, they produce high levels of proteins like p53, which deactivate the cell growth arrest, allowing more cells to progress with damaged DNA, turned into oncogenes which proliferate extensive cell growth rather than reaching apoptosis.
For so long, many people only believed melanoma was cancer caused by increased exposure to ultraviolet radiation from the sun. Familial melanoma is a genetically inherited condition that acted on the CDK2NA, CDK4, and P53 genes in the DNA, which is important in initiating cell division and increases susceptibility to melanoma development. Familial melanoma patients are diagnosed at an earlier age with thinner tumours and a higher frequency of melanoma. P53 is a tumour-suppressing gene located on the short arm of the 17th chromosome and codes for 53-KDA phosphoprotein, which occurs in gene transcription and causes catastrophic consequences to the cell cycle. CDK2NA is coded for cyclin-dependent kinase inhibitor 2A and is also a tumour-suppressing gene, which means that they keep cells from rapidly dividing in an uncontrolled way. CDK4 stands for cyclic-dependent kinase 4, which controls progression through the G1 phase of the cell cycle. Unlike the CDK2NA gene, this gene plays a minor role in hereditary melanoma and is mainly caused by excessive exposure to UV radiation.
Colorectal cancer, breast cancer, ovarian cancer, and melanoma are some of the many cancers, but this could be your reminder to get checked.